Thank you everyone for joining us for today's CCDS community chat. We're excited to delve into CCDS research with our research advisor, Sangeetha Iyer. Before we get started, we'll just do a few housekeeping items. Today we are initially meeting in webinar mode, and we will get through this information and slides, and we'll check in on the Q&A. So if you have any immediate questions you'd like us to discuss, we'll do that. And then we'll conclude the presentation portion and end the recording, and we'll have a more open discussion with all of you coming off camera if you choose to at the end. So just a quick heads up on next month, December 13th, our community chat will be combined with the parents group. So we've got 28 caregivers that are attending monthly meetings in our Parents 2.0 program. And next month, they are learning about the drug development process. And so we are going to combine that with our community chat, invite all of you to be a part of this. And we are offering that meeting at 2 different times. So you'll see 2 calendar invites. Just choose the one that suits you best, noon or 5:30 Eastern time, and we'll get that link out following this meeting. And of upcoming importance, we are launching our Holiday Heroes campaign that we have run every year for 12 years now. On December 3rd, that's Giving Tuesday in the US, that's the Tuesday after Thanksgiving. And we're asking all of you to sign up and fundraise alongside with us. Hopefully after you hear about the research progress that's been going on and where we hope to go with research in the future as quickly as possible to find a cure for our children, you will see the importance of this. But I also have a pretty big announcement. This QR code, by the way, is where you will go to fill out a form to sign up to participate. I will tell you it's really simple, and we make all of the materials for you. You take the materials and you post them once a week or so, just telling your friends and family about the campaign and the importance of this research for you. And our announcement is we have our biggest individual donor ever pledged for $125,000 from a generous individual who would like to give a Bridge of Confidence gift to our community. A Bridge of Confidence gift is where they say they want to see that our community is actively committed to this, indicating we're going to put these funds to use. We need these funds. We're committed. We're active. And that's why it's dependent on our community raising the first $200,000. So for reference, we have exceeded $200,000 for the last 2 years. So that's our first fundraising goal. Overall, our fundraising goal for the entire community is that we ourselves, before that Bridge of Confidence gift, raise $300,000 so we can do all the things that Sangeetha is going to tell you about. So again, if you haven't signed up yet, please do so. We're creating a lot of materials that will feature the pictures and the videos that you all are submitting, and the more families and affected patients that we can have, it's just that much more impactful and powerful for the people we're asking to donate and to care about this cause. So please join us. It's very simple. So with that, I will turn the webinar over to Sangeetha Iyer, and she will share her slides. I'm going to make her the host, so stand by for one moment.
Okay, I think I turned on my video. Can everyone hear me, see me? Excellent. Give me one moment as I bring up my slides. Okay, can everyone see this? Let's go maybe to the start. Okay, all right. Well, thanks for joining, everyone. I'm looking forward to interacting with you all today through this a community chat series. And what I really wanted to do today is maybe present a bit of an overarching landscape of therapies in general available to all disorders, and then focus a little bit on the landscape, therapeutic development landscape specific to CCDS overall. And what I'd like for you all to do is to I'll go through the deck, but as and when possible, please pop in some questions. Heidi, perhaps you can help me out in, if there's a question in between that I should answer, then if you can flag that for me, that would be great. And yes, I really like to get into a discussion closer towards the end on any additional questions that you may have that may have not been answered by the content here. Okay, but before we get started, I thought first I'll maybe provide a little bit of my own bio. I know that you've seen me, the majority of you guys have seen me at different conferences, and I've certainly been affiliated with ACD for a number of years now, but I wanted to just take a moment to dive into my background. So I can provide a little bit more context for the content that I will be sharing today. And also for the questions that I am more specifically equipped to answer, and some might just be out of my purview. So with that prelude, what I wanted to say is that I have a degree in molecular pharmacology from the University of Pittsburgh. I started my career about both academic and extra academic career about 15 odd years ago. After my degree in pharmacology, which was primarily focused on genetic engineering of mouse models, I completed a postdoc at UT Austin. Through my PhD and my postdoc, I work largely with different models of disease, be it cellular or more along model organisms, whether it's mouse or more basic organisms such as C. elegans and Drosophila. Large part of my work was actually studying ion channels. I studied GABA, glycine, and BK ion channels. All of these channels are particularly important from an epileptic perspective, and certainly important and implicated in a variety of neurological conditions. Subsequent to my academic training, I started my career in a small startup that was focused on small molecule drug discovery for rare diseases called Perlara. And I primarily joined the company and worked on drug screening, and development of different in vitro or cellular models as well as animal models of rare diseases. And I worked on these particular disorders over while I was at the company: Niemann-Pick type C, and glycanase 1 deficiency, and phosphomonomutase 2 deficiencies, all of which are fall under the category of rare or ultra rare. During my experience over there, we were fortunate to actually discover a small molecule modulator of an enzyme deficiency, which is PMM2. So this figure that you see over on the right over here is a figure of Holly Carmichael and her daughter Maggie. Maggie has PMM2, and we modeled her mutation in the fibroblasts, and worm models that we were utilizing at the time found a molecule that actually increased enzyme activity, and there is now a clinical trial underway leveraging that particular discovery. And to put in context, the discovery around the PMM2 molecule, the molecule that we discovered, that discovery was made in 2019. And phase 3 clinical trials are now underway, and we are in 2024. So even after discovery, as I'm sure everyone on this call is familiar, many things happen. Happy to go into that story in greater detail if there's interest. But that was my background with rare disease small molecule discovery, and one of the big reasons for why I was really motivated to work in the rare disease space, and particularly with patient advocacy groups, and why I have a bit of an appreciation for the systemic lack of tools, reagents, and the like that is required to bootstrap early discovery efforts. And maybe a bit of a perspective on the different types of incentives and opportunities that are available to those who develop drugs in the rare disease space for what kind of paths they might pursue to move something towards a clinical trial. It is definitely not a one size fits all, as you can well imagine. Subsequent to that, to my experience at Perlara, I spent a couple years at Denali Therapeutics, where I appreciated or got to appreciate more of the large molecule or biologics discovery and development pipelines. And then I transitioned after that role into portfolio and partnership-based roles at Pfizer and my now company, Big Hat Biosciences. So I worked across different therapeutic areas in small, medium, and large pharma companies, both from a pure technical discovery perspective, as well as an overall portfolio strategy and partnership strategy perspective. And it's that vantage point with which I work with now with the ACD to understand what are our most pressing needs on the research side, as well as on our strategy for what are the kinds of things that we may want to pursue, what are the types of partnerships that we want to engage in that will take us to the next step, closer to cures, certainly for the community. Okay, so that was just a little bit of background about myself, and now I want to start with a brief primer on the state of therapeutics, right? So what I really thought I would do is maybe compare and contrast for this group what small and large molecule therapies are, and what's the status of small molecule therapies and large molecule therapies. What are some of the challenges and some of the maybe advantages even of pursuing one over the other. And so here what you're seeing displayed on the screen here, this Y-shaped molecule is an antibody. It's an antibody therapeutic. And it is utilized for breast cancer. On the right over here is a very common molecule, Lipitor, which is used for hypercholesterolemia. The thing to note over here is that the size of this antibody is 185 kD or 185 kilodaltons, whereas the size of the small molecule is really about 500 or 0.5 kilodaltons. So one thing to appreciate straight off the bat between large and small molecules is the distinct difference in size. The other thing to sort of keep in mind is really what pursuing a small or large molecule means for later stages of development. What are the types of challenges that are encountered in taking these molecules ahead towards clinic, right? And so as I just shared, for small molecules, you'll see that there's a distinct size difference. The structure of small molecules is simple and defined. You can manufacture these by chemical synthesis. They're relatively stable. They don't undergo a lot of changes. And once, for the most part, from a toxicology to the immune system perspective, small molecules are typically non-toxic, mainly because they are cleared from the system or they're eliminated from our bodies on a regular basis. By that same token, they also require more frequent dosing. So you can imagine that small molecules need to be dosed every day, maybe multiple times a day. And that usually is in contrast to a large molecule. The other thing is that from a quality control standpoint, because you can synthesize large quantities of a small molecule because the mass is small, you can have greater stability, or greater identical copies of the same made. Now, in the case of biologics, this is a little bit different. So as I already shared, this particular large molecule is a biologic. Uh, the size is much larger. It usually has a complex and heterogeneous sort of manufacturing process. So it's not put all ingredients, put all basic materials in a pot, and raise the temperature, lower the temperature, and you get a predefined element over there. Most biologics are made in living cells or organisms, and then beyond that, they may or may not be coupled with some kind of non-biological complex, for example, say nanoparticles. So you can have nanoparticles encapsulating a biologic material, such as a vaccine or an mRNA or gene or antibodies or the like. Okay, and then there's many different modifications that you can now make on antibodies that enable better targeting, very, very specific modes of action, very, very specific deliveries to certain targets of cells and the like. However, because they are produced by living cells or organisms, usually a mammalian cellular system of some sort, or a bacterial, in certain cases, they can be bacterial cellular systems. There's much less control we have on influencing the yield or influencing the batch to batch identity of certain types of materials. And because these are complex, large molecules that may or may not require a downstream step of integration, they can be unstable and sensitive to external conditions. And you'll note this that, you may have heard about this for the mRNA vaccine, that it was able to be transported only in -20 degrees Celsius, only in certain types of vials, and it needed to be reconstituted in a certain way before it could be administered. And that's very much true of the class of molecules of biologics. Typically, biologics are not administered at home. They will be administered in some kind of hospital or other outpatient setting. The dosing of these types of large molecules, because they're so complex and because there are stability and other constraints, the dosing for these is usually handled by an experienced individual. So some sort of clinician or some clinical caregiver, as opposed to allowing you to go home and take it by yourself. And by that same token, it is administered less frequently. So these biologics are not usually given every day. Then ideally, you wouldn't sign up for a weekly dosing. You may sign up for once in 2 weeks, once in 4 weeks, or once every couple months. Or if it's gene therapy, then maybe only once ever. The other thing to note for the development, like a compare and contrast in the development lifespan for biologics, is that because these are molecules that are generated from living cells, and because they have very, very specific properties that are engineered into them, so that they can be supported by this dosing frame of only once in every so many weeks, they can be immunogenic. So that is to say that they have complexes in them that are derived from another organism, and as such, those particular modifications can trigger immune reactions from our bodies. So when they are administered to us, either the core molecule, the biologic itself, or in certain cases, the complex within which it is encapsulated, say a nanoparticle or a vector of some sort, those can give rise to immunogenic properties within us. So the liability for some of these biologics in later stages of development is really how they perform in clinic. Do they support a good dosing window and very low adverse effects, or is that something that we can sufficiently de-risk? And because they're produced by living systems, last note to compare and contrast is that, like, it's a little difficult to ensure identical copy versions, and this is true for like gene therapy batches and the like as well. There's a ton of work that goes into ensuring that exactly the same number of copies of a particular gene or a particular viral vector are dosed patient to patient. When you have batch to batch synthesis of these, then there are special hoops, special considerations, special QC checks that you have to pass through in order to ensure things like that. Hopefully that was a reasonable overarching view for how these two different modalities differ. Moving ahead, next, what I wanted to do is, before we get into the landscape of therapies for CTD, is talk a little bit about small molecule approaches for CTD. So this schematic is from the Sonia Suchik paper from several years ago. And what it does is to try to outline. They published a paper actually on pharmacochaperoning, but this is a very nice schematic that actually outlines what are the different types of approaches that you can take for CTD, and really generalizable to most transporter strategies. So what you're seeing over here is a typical cell. This particular component in the inside of the cell is the endoplasmic reticulum inside of the endoplasm, and this is the usually the cytoplasm. And in the cytoplasm, you have numerous organelles. Like you have the endoplasmic reticulum, you'll have the nucleus, you'll have the Golgi apparatus, you'll have the ribosome, you'll also have lysosomes, and a couple things that are not represented very specifically over here. Now, typically for any protein, a protein is usually translated. So the nucleus makes the mRNA, the ribosome makes the translation or the protein, and then it usually goes into the ER as well as the Golgi for additional maturation. And one of the places where quality control takes place is the ER, or the endoplasmic reticulum. In the endoplasmic reticulum, there's a bunch of different types of proteins that are there that determine whether the protein is folded properly or not. And if it is folded properly, then the protein is allowed to exit the endoplasmic reticulum, enter the Golgi for additional post-translational modification, and from there into its final destination, either as a soluble protein. So for context, enzymes are soluble proteins. They are usually synthesized, put through this QC check, and then they're floating in the cytoplasm, for lack of a better word. And in the case of membrane transport proteins, they are usually, after undergoing post-translational modifications, they are usually inserted into the membrane of the cell itself. And so, based on the process of how a protein matures after translation all the way to insertion in the membrane, there's a number of different ways where you can intervene for ensuring better translation or expression of the protein finally. So here you can see what happens in the case of CTD. You don't have the transporter protein because it is bearing some kind of mutation. We think that there's a large majority of proteins that are usually stuck in the ER, may never make it over here because of QC checks and the like. So one of the ways with which creatine transporter disorder is thought to be resolved is pharmacochaperoning. And here you're leveraging a bit of you're leveraging the drug itself, so something like 4-phenylbutyrate, or you're leveraging a pathway associated with natural or endogenous chaperones, or both. So heat shock proteins, for example, are ones that are highly expressed in the case of misfolded proteins, and what they do is essentially step up the quality control function of a cell. And by modulating or tweaking how these heat shock proteins act, you can change up the process of quality control. In the case of CTD, the intent would be to make it less stringent, more permissive, to allow expression of a sub-functional transporter. Okay, and so what you are seeing over here is a little bit like that. This is an example of a SLC6A8 transporter bearing a mutation that has been subjected to some kind of pharmacochaperoning treatment, which allows it to escape quality control and increases its expression on the surface, allowing it to transport creatine at whatever level of function is salvageable. that might be 30%, 40%, or lesser, but it may still lead to more creatine in the cell relative to not having it there at all. This particular schematic talks about leveraging a different transporter, and so there have long been these ideas that if you can conjugate creatine, which in and of itself is a relatively small molecule, if you can conjugate it to another small molecule, and by that I mean small nutrient molecule that is required by the cell, then it could utilize a transporter that is associated with the other nutrient. So case in point would be something like a glucose, or that requires the glutathione transporter, which I believe has been tried before and was not found to be successful. But there may be examples of other transporters, lysine, other amino acid transporters that creatine could be conjugated with, and then you could essentially use a decoy approach to bring creatine in using another transporter. The thing to note about this type of approach is that other transporters also have relatively stringent QC checks. So you cannot make a molecule that is super different than the one that it is likely to recognize. So identifying this other transporter, or what we may call as a bypass transporter, can be a bit of a challenge. But that's an approach that we at ACD have not explored and would like to explore. We are still searching for the right subject matter experts that can actually enable us to pursue something like this. Another method of bringing creatine in, and this is one that has been pursued, is using a lipophilic creatine analog that does not require any transporter. So if you conjugate creatine to fatty material like esters, or the like, then you are able to, because the plasma membrane is usually made up of a bunch of fats, it's a little bit more tolerant to a fatty material. And so in this case, this type of material does not require a transporter to get in. It requires insertion into the plasma membrane, and then it gets in. The one of the challenges associated with this method would be that because you are looking to passively insert creatine with some kind of lipophilic macromolecule or lipophilic structure, you don't know how long it will take to get the creatine in, and whether you can actually achieve high enough concentrations as is required by the cell to convert the creatine into phosphocreatine and downstream into its energy metabolics. Last but not the least, and this is an approach that we have tried a little bit, is whether you can empower a cell to synthesize its own creatine. So creatine is actually made from using these precursors of arginine, glycine, GAA, and it requires the use of enzymes such as AGAT and GAMT. Those are the biosynthetic enzymes. And now we know that in the brain, there are certain cell types where GAMT is not present, and instead those cells have SLC6A8, because under normal situations they can bring in creatine externally. But in the case of where you have mutant or you have a non-functioning transporter, if you could confer that particular cell with the ability to take a GAMT and give it the right substrates and enable it to make the downstream creatine by itself, you have now made that cell type a de novo producer of creatine, because it has everything that it needs to produce creatine internally. And that way it no longer needs to rely on the transporter to bring external creatine. So that's the other way, and one that we have explored as well briefly. Okay, so let's talk now about the landscape of what's been done in the field of CTD itself, right? And I have a few slides on GAMT as well. So Lumos Pharma was the first one that I'm aware of that had a CTD program. They had a cyclocreatine molecule. So this is an analog molecule that basically was thought to improve cognition in mice bearing CTD behaviorally. and this dates back to about seven odd years ago at least. Unfortunately, they had subsequent, that was the cyclocreatine was a molecule that they were planning to take ahead to the clinic, and they did do a number of additional preclinical studies in order to understand dosing as well as toxicity profiles of the molecule, and they came up with the finding that cyclocreatine, for unknown reasons, was toxic in wild type animals. So you saw in this previous title, that they actually studied the treatment in mice with CTD and saw improvements in cognition. But when they gave the same molecule at higher doses to a rat model, normal rat model, they saw evidence of toxicity. They followed up this with evaluations in beagles as well, which is a common species utilized for GLP toxicity, or uh, to this, there is an aspect of drug development where you have to assess two species toxicology, and rat and dogs are typically, or mini pigs are in some case, utilized in those types of studies. This molecule went far ahead before they identified that there were toxic issues associated with the molecule. And what you're seeing over here is a survival plot of treatment in dogs. And so this plot, this top bar over here, you guys, you might not be, are you able to see my mouse? If not, let me see if I can.
Yes, we can see your mouse.
Oh, perfect. Okay, so this is the control line. So these are animals that did not receive the molecule. They all survived. And then you can see this is the low dose, the medium dose, and the high dose. And this is telling you what the survival ratio was. And so there was a dose dependent toxicity in these dogs, resulting in their premature death, that was related to very clearly related to the dose itself. And just for a brief bit of compare and contrast, I do want to say that usually the doses that these types of studies are done at are relatively high, and it's not expected that for efficacious drugs, for drugs that you see on the market that you are taking and are working for you, those doses are usually much lower than what are studied in this type of study. But this type of study is done to establish that therapeutic window, to establish how much of a separation do you have between the dose required for an efficacy or effect, positively affecting the disease, and what's the window from that highest dose to a toxic dose. And if that window is lower, then you say that the therapeutic index of that drug is lower, and that presents a problem or a complication for individuals who need to take the drug for positive effect, because that means that there isn't any window. So this program was terminated following this finding, and cyclocreatine since then has not been evaluated for CTD. As everybody on this group, on this call is likely aware, Ultragenics subsequently took over the natural history study and had their own program for CTD. Their lead small molecules was UX068. This is data that was, we've seen different versions of these data over the past year. This is just to tell you that from the point of view of where does it fall, what kind of therapy it is, it's a small molecule prodrug. So utilizing some kind of lipophilic structure that is essentially able to pass the blood-brain barrier, small enough to pass the blood-brain barrier, small enough to insert into the cells and actually release creatine internally. In 2019, they were expecting to file an IND. In 2020, we since know that was not the case. They had to go back and reformulate the molecule to make it amenable to oral dosing. And we have not since heard from them. What they did show based on preclinical studies, efficacy studies, is that they were able to show a distribution of creatine in SLCA8 knockout rat brains. And this is just imaging data showing that following dosing, 24 hours after dosing, they were able to see widespread distribution of creatine in the brain. From these types of figures, it is usually not very easy to tell exactly which cell types it is in, but as you can see, this is the knockout brain, and this is kind of all over, has no creatine, and this is getting relatively close to wild type creatine. So very promising molecule. However, we have since not heard from the companies. We don't know if the molecule has been stopped in development because of strategic reasons in the company, or if they're still reformulating, or if there's other liabilities that they have uncovered because of which it's not going ahead. And then moving forward, we have obviously heard of Ceres as well. Ceres is also using a prodrug mechanism, a lipophilic creatine analog, in order to deliver this molecule through an inhaled route of action. So the route of administration over here is different from Ultragenyx. The route of administration here is inhalation, and therefore will be capped by volume. In the previous situation, the earlier version of the formulation was delivered by injection. I can't quite recall whether it was subcutaneous, intravenous, or the like, but it was some mode of injection. And because creatine is something that is required every day, you can imagine why something that is an injection maybe multiple times a day and every day may not quite be suitable for dosing in our population. An inhaled route in principle might be somewhat easier, although the proof is in the pudding. It depends on how frequently you have to take it and how much actually gets in based on that. But all this to say that, just to relate it back to the figure that we were looking at, there's two approaches that are looking at creatine analogs thus far. This one also is somewhat of a complex molecule because here the creatine prodrug is encapsulated in some version of nanoparticle, but a relatively simple one as we understand. Okay, I'll keep going. Janana Therapeutics was another company that was also working on SLC6A8. They were leveraging the pharmacochaperone methodology. And this paper came out recently. However, they also shared some data with us in 2020 that I'll call your attention back to in just a minute. But this particular paper talks about their screening platform and the technology that they used to identify molecules that could stabilize many different point mutation bearing variants and enable an increased expression on the surface of the cells. And this is what this particular figure is showing. They have multiple variants and one corrector molecule, and across many different types of variants, this particular corrector molecule was able to increase the surface localization. You're also seeing that in certain types of variants, this molecule was able to increase the creatine transport. Creatine transport to wild type, or in certain cases exceeding wild type. So 100% would be wild type, and so more than that means that the molecule was able to, for certain variants, definitely restore and go above and beyond in terms of encouraging creatine transport. What the paper didn't really focus on, but what we heard in 2020 was their lead molecule did have toxicity. So this is the data that was presented at our symposium in 2020, and you can see that they tried many different types of assays. They looked at the compound levels. They looked at the compound levels with twice a day dosing, once a day dosing, and they were able to show that you are able to get creatine concentrations in the brain in these particular mutant mice, the P544L mutant mice. These are heterozygous female mice. And then they also showed the hemizygous and wild type male mice and the residual creatine in one particular area of the brain. This lead compound basically did increase brain creatine across different types of regions of the brain. However, it was not tolerated. So 30 milligram per kilogram was not tolerated past 3 daily doses. They terminated the study due to poor tolerability, and it also ended up with them dropping the program. Compare and contrast against the Lumos study, as that this one was done in the P544L mutant mouse model, whereas the Lumos tox findings were identified in a wild type rat model. The P544L has been discussed in the past as having some residual creatine transporter activity. So what's not clear to us in the field, based on these types of data that are coming out, is that whether there is a certain level of creatine above which, if we go by using some kind of synthetic mechanism, the brain is not able to tolerate it. There's toxic liabilities associated with it. Janana also acknowledged that the toxicity that they saw, and this has also come up in the context of cyclocreatine, that it may just be molecule related. So it has nothing to do with creatine, nothing to do with how much more creatine it enables, but has everything to do with the molecule itself and the way it is handled by the body, by the liver, by the kidney, or other reasons. I don't believe that anyone knows the answer to this in the field, but this is one of the things that we will be keeping a close watch on as these small molecule therapies come to fore. Okay. So the Janana work previously, as well as work from the Suchik lab, essentially indicated that chaperone correctors would work on point mutants mainly. So there was a bunch of in vitro data that was collected by the Suchik group, and also obviously, as you saw, Janana, where they mainly focused on point mutations. They were not able to show corrections in an in vitro model system in point deletion mutations or large deletion or duplication based mutations. However, particularly with the case of 4-phenylbutyrate, it's not really clear. 4-phenylbutyrate is sort of a transporter agnostic or protein agnostic chaperone, which is to say it works on the chaperone machinery of the cell rather than being specific to any given transporter. In Janana's work was geared towards finding a chaperone that was specific to SLC6A8. So there is probably some element of binding of the molecule to get the molecule getting into the cell, binding to a misfolded version of SLC6A8 and somehow stabilizing expression. Relative to something like 4-phenylbutyrate, which has been known, and we have come to also understand that it works on multiple types of disorders. So this is evidence that was stated by or published by Dr. Zachary Greenspan before he approached our community for a study on CTD patients with 4-phenylbutyrate. So as you can see, there are two different disorders that are represented in this table, 6PB1 and SLC6A1. And they are both, in the case of both of these types of disorders, 4-phenylbutyrate was able to correct the disorder. I need just one moment, if you'll excuse. Apologies. Okay, so getting back to the table over here. So across two different types of disorders, in a mutation agnostic manner, 4-phenylbutyrate was able to produce some correction. What is not clear in this particular case is if the correction was related to just seizures, so is working in some manner that is more relevant to seizure pathology, or whether it has anything to do with the mutated protein in question. SLC6A1 is also a transporter disorder. Between that and other studies, there's reason to believe that it could be doing something at the protein level. However, the end point that was corrected in these particular disorders were seizures, which is why Dr. Greenspan was looking at other seizure disorders in which to extend this type of finding or study to. And because a certain proportion of CTD patients also have seizures. Seizures was essentially a precondition for joining the study. However, type of mutation was not. I hope that maybe clarifies certain things or provide some context for how this study came on our radar. Okay. I'll switch gears very briefly while on the topic of small molecules to also talk about GAMT deficiency. As most everyone in the community is aware, in the case of GAMT deficiency, we have had the mainstay of therapy has been supplementation and diet restriction without really any small molecule or biologics, large molecules to really that are available now as solutions just yet. And so there are two paradigms or a couple different paradigms that can be thought of for small molecules for GAMP deficiency. And I will talk about one today that has been evaluated and perhaps shed light on what else could be evaluated. If you are familiar with the biosynthetic cascade associated with the production of creatine, AGAT is the principal enzyme that works on arginine glycine to give rise to GAA. And then GAA is acted upon by the GAMT enzyme along with another nutritional supplement to give you creatine. And because the systemic lack of creatine in GAMT can be resolved by giving those children creatine, what the lack of GAMT does is it allows a buildup of toxic GAA, which is thought to be excitotoxic and results in chronic neurotoxicity. So the seizures that you see in GAMT are associated with the buildup of this chronic neurotoxicity. The other thing to know is that when the brain is continuously exposed to a continual basal level or even elevated levels of a neurotoxin, hyperexcitability sets in, and hyperexcitability can result eventually in neurodegeneration. And that's the reason why even with supplementation and other things, because you're not resolving GAA, sometimes you don't see resolution of seizures because the brain is still exposed. Unless you have very tight control on diet, you're not able to resolve GAA, and continuous exposure of the brain to GAA is considered to be, or to any excitotoxic molecule, is considered to be chronically bad. And so one of the methods to consider resolving GAMT is thought to be an inhibitor of the AGAT enzyme so as to reduce the amount of GAA that is produced. And so this was evaluated, certain molecules were evaluated by Dr. Longo, and these molecules were generated by this company using an in silico approach. The company was called Atomwise, and this is data that was presented by Dr. Longo again at one of our symposiums a couple years ago. I want to say 2023. What you're essentially seeing over here is that there are molecules that essentially reduce the amount of GAA to what is considered to be relatively low levels. So these are the different types of molecules that were tested in a 96 well plate, and these are the GAA levels corresponding to those particular molecules. And here you can see that when you have positive controls of the enzyme itself, you don't have any GAA at all. Right, so when you have functional GAMT enzyme that is able to utilize all of the GAA, you don't have any residual enzyme in the multi-well plate. But in the case of these small molecules that are inhibiting the enzymes, you are seeing some amount of GAA being produced, but there are a number of molecules, as you can see this one, so 1 and 2 and maybe 3 over here, that are able to reduce the levels of GAA. Unfortunately, the conclusion after doing the study from this paper was that the concentration at which these drugs needed to be used in order to lower the GAA levels were very, very high, meaning that it would not be suitable for future development, because If I remember correctly, they were micromolar quantities, double-digit micromolar quantities of the drug that were needed to produce this type of effect. And lowering the concentration of drug did not produce a strong inhibition of GAA, which meant that chemists are essentially back to the drawing board in being able to iterate on these molecules and make more potent versions of them. So what is the lowest amount of drug that will be required in order to drop the GAA levels? So when you get to from an in vitro assay standpoint, which is to say cells in a dish, if you have molecules that are in the low micromolar or nanomolar ranges of concentrations, that usually supports a path for future development, which means that it can eventually be compounded into some sort of a pill. If the starting concentrations are very high, then it does not usually support a path for development, because the quantities of drug that will be needed would be very, very high, and not sustainable with dosing. Okay. Now we'll switch gears to large molecules. Heidi, I'm going to keep going unless you give me a signal to stop in case of questions or the like. So I will wait for a signal from you, and otherwise keep going. But just let me know if you'd like me to pause and expand on something.
No,
Okay.
I think we're doing great, and there's no questions in the Q&A. So
Okay, awesome. So I'll switch gears to large molecules. So we are at a base level somewhat aware about what large molecules are. There's probably two categories, not like really strictly demonstrated on this particular table, but in the context of large molecules, any large molecule other than an antibody, typically antibody or enzyme really will require some kind of vector or material in order for it to be transported. That's the case in the case of gene therapy, or in the case of any other nucleic acid such as mRNA. Certain types of antibodies can also require nanoparticles or some kind of complex macromolecule for delivery. But we've reviewed some of this in the previous slide. But the idea is that they are derived from the large molecules are usually derived from some sort of biological origin. Their path to developability is very high, specifically in the case of, path to developability is very long, specifically in the case of gene therapy. There's a lot of different types of parameters that needs to be taken into consideration for effectiveness. So when you consider viral vectors for gene therapy, you need them to be transfected at a high enough number. Non-viral vectors will show you lower degrees of transfection, which is to say that the amount of packaging that something can take will be limited. So we're talking about this packaging capacity. In the case of gene therapy, you have very small quantities in terms of the size of the gene itself and the number of copies. There's a limit to that. In the case of non-viral vectors, you don't have a size limitation, and non-viral vectors such as LNPs can certainly accommodate mRNA or protein or any really type of cargo. And then the other part of this is that if you are looking at viral vectors such as an adeno-associated vector for gene therapy, you do have the possibility of immune reaction. You have the possibility, because this is true of any viral vector dependent delivery, that components that you are sending in with the viral vector can integrate into your DNA. We can talk about this a little bit more, but for those in the audience that follow the gene therapy field significantly, there was recently a paper associated with gene therapy with a late developing side effect associated with gene therapy for X-adenoleukodystrophy. So about 3 years after the gene therapy was delivered, certain types of cancers were seen in those in 7 out of 60 odd patients that had received this gene therapy, and that was thought to be associated with viral vector based integration of the DNA. And it happens randomly, or it may have a preference to certain loci. So it's not entirely clear how that happens, but that possibility exists. In the case of non-viral vectors, because they are quiescent at some level, dormant at some level, LNPs, they are less likely to deliver, to elicit immune reactions from the get-go. There's less likely because they're not derived from a live component, so to speak. They're just complex and large molecules. They're not derived from a live entity of any sort. They have fewer elements that can cause insertion into the DNA. However, they have the problem, LNPs and the like have the problem that they don't integrate, they don't deliver to all types of cells. Certainly, we have not been able to target LNPs into the brain with any success. We are only able to target LNP to livers with a high degree of success. Technologies that enable tissue targeted delivery of LNPs are underway, but I'm yet to see a clinical trial that has something super specific for any organ other than the liver. Okay. So when on the topic of gene therapy, I want to say that we've certainly seen gene therapy for GAMT deficiency from two different groups in our community. So one was Dr. Lipschutz's work, and the other was work from Dr. Valia's lab, which you would have reviewed in, or we had an access to, or had an opportunity to listen to if over the past 2 years at both of our virtual conferences, virtual and in-person conferences. Both of these groups have demonstrated correction in the preclinical, so mouse models, with GAMP gene therapy, and both of these groups are advancing this gene therapy to the next phase of development. So generating larger quantities of it and putting it through the next necessary toxicity and other studies before scaling to clinical development. Dr. Lipschutz in particular has received a large grant for his gene therapy work, and this is really exciting times for our community because there may be a GAMP gene therapy on the horizon, provided it goes through the appropriate safety and toxicity de-risking steps. Similarly, there have been definitely a lot of studies into gene therapy for creatine transporter deficiency. However, not all of these, while they have been successful in correcting certain phenotypes, I would say that the goalpost is a little bit further off for CTD, because we need better understanding on which are the cell types that need to receive the gene therapy. What is the consequence of delivering gene therapy to multiple cell types, particularly being aware of some of the small molecule findings and certainly early findings from gene therapy for SLC6A8. We need to understand the landscape of toxicity associated with creatine transporter function and with higher levels of creatine itself before we can say we understand everything and we think that this is ready for clinical development. But for this group, I think that you all should be maybe happy, and I think it's important that gene therapy as an option be evaluated at all, and certainly you should feel a certain amount of pride because it's your efforts, your work in fundraising that have enabled these studies to come to fore. This is especially true of actually all of the groups that we have just spoken of, whether it is Baroncelli, whether it is Lipschutz, the Bresant lab, or the Valia lab. All of these elements have been initiated because of our engagement and certainly some of the avenues of funding that we have been able to create. Okay. I do want to talk about, now that we've spoken about gene therapy, don't want to miss the opportunity to also talk about enzyme therapy. And as you know, GAMT and AGAT are both enzymes, and definitely there have been numerous instances of ways to leverage enzyme therapy for rare enzyme disorders. One of the ways to achieve this has been to reduce GAA. So for GAMT, there can be two avenues for resolving disease. One is that you try to replenish GAMT, or the other is you try to again reduce the levels of GAA. And you can reduce the levels of GAA either by inhibiting AGAT, which we saw on the small molecule approach. The other way to inhibit the production of GAA is to produce an enzyme that takes away the building blocks of GAA, and that is pegylaarginase. So pegylaarginase is a molecule that's actually, it's a large molecule or an enzyme, synthetic enzyme that is being developed for arginase 1 deficiency. It was originally developed by Aglia Bio, actually developed for cancer. And then it was taken over in the past year by Immedica, which acquired the asset and is now moving forward for approval in EU for arginase 1 deficiency. We have spoken to Immedica and researchers that were involved in this type of study to extend it to GAMT. It's early days yet, but we are hopeful that we can engage with them in a concrete manner, that they will agree to take this type of molecule and study it in GAMT patients as well for the purposes of reducing GAA. Just last couple slides, I promise, just to shed some light on what are other things that are on our radar that we want to pursue. So this here is, I've already spoken about pharmacochaperoning. We've seen about how pharmacochaperoning can actually be utilized for CTD. The same is actually true for GAMT. And I've spoken about this a little bit in the past. GAMT arises due to compound heterozygous mutations. So, or it can be homozygous mutations as well. So homozygous mutations are mutations where you have the same defective copy on both alleles, one from each parent. Heterozygous mutations are where you have received a different mutation on the GAMT gene from each parent. And in this case, net. So cumulatively, both of the mutations result in a defect in the gene, and hence there is subfunction or no function of the resulting enzyme. What we have not studied adequately in the case of compound heterozygous mutations is that if one of these alleles has any function that can be leveraged or that can be rescued, then you could potentially leverage just that one allele and enough copies from that one allele in order to be able to correct the disorder. I say this with a little bit of experience because that is what we found in phosphomonomutase 2 deficiency, which was also an enzyme disorder, also arising due to compound heterozygous mutations. The individual in question had one extremely deleterious mutation and one subfunctional mutation. The subfunctional mutation was amenable to rescue by a molecule, and that's how enzyme activity was somewhat restored. We have not studied this in adequate detail in the context of GAMT, and I do feel like there is an opportunity in the case of compound heterozygous mutations to do the diligence to understand if there is at least one rescuable copy of the allele. So that's something that we've been looking at opportunities to fund. This is more along the lines of straight up enzyme replacement therapy. So we've looked at gene therapy as an option for GAMT as well as SLC6A8, for AGAT as well as GAMT, because these are enzymes and GAMT in particular is produced in the liver. There is an opportunity for us to, for someone to actually deliver synthetic GAMT enzyme to the liver and see if that can be utilized to rescue the disease even peripherally. There is an argument to be made for, like, yes, there's GAMT also in the brain in very specific cell types, so just resolving the disorder in the periphery may not be sufficient. So delivering enzyme to the periphery may not be sufficient. But also I'll make the counter claim to whoever gives me that claim that I don't know that we know that 100%. I don't know that we have tried resolving GAMT in the liver and determined that it doesn't drop the threshold of defect in the individuals with the disorder, or even mouse models. We've not successfully evaluated this in great detail, and this is another area that we're looking for groups that specialize in making synthetic enzymes and delivering them to mouse models. We're looking for groups that will be able to do this work. And so this, along with the previous pharmacochaperone approach, is an element of focus for us going into next year. And then I put this slide mostly for folks because every so often we will receive emails on like, mRNA vaccines were developed really quickly. What's the opportunity for us? Have we looked at this in significant detail, right? And this is just a landscape of therapies that we have not, truthfully speaking, we have not looked at in great detail. We have not looked at CRISPR-Cas9 based mRNA editing or CRISPR based gene editing or mRNA. And that's because a lot of these are newer technologies. The path to developing them to clinic is not adequately de-risked. We don't even know all of the problems that are associated with these types of modalities. mRNA in particular, I will say, might be an option for GAMT or enzyme disorders, because in those cases, mRNA has to be delivered to cell types and result in a soluble protein. Whereas in the case of delivering mRNA to make SLC6A8, you are looking to deliver exogenous mRNA that will produce a protein that can be folded properly and inserted into the membrane. This has not been demonstrated in the context of mRNA. It has truly not been demonstrated in the context of gene therapy either. But in the grand scheme of things, mRNA is a newer technology relative to gene therapy, and for gene therapy, we at least know something. We know what we don't know, at least for gene therapy. I'm not sure that the same can be said about mRNA because it is newer in my mind. That's not to say that we won't ever evaluate it in the future, but in the interest of building, balancing the build of a diversified post portfolio, which has some de-risking, we're placing a mix of calculated bets. And that's why we haven't pursued mRNA as significantly. We're keeping an eye on evolving technologies in this space. The same is true of CRISPR and Cas9 gene editing therapies as well. That these are all relatively new, and the idea of delivering CRISPR-Cas9s to engineer very specifically a locus, a specific site of mutation for SLC6A8 or GAMT. This is not on our radar currently because this has been demonstrated successfully in the preclinical space for a number of disorders. There might be some early bets being placed in clinic for larger indications, and we should look to those studies to understand what kind of safety risks there are, what kind of efficacy profiles there are before we pursue those for our disorder. And with that, I want to end on a plug for or maybe a brief review of our fellowship program and plug, right? Which is that a lot of what you saw today was based on, particularly the ones that are not in corporate development efforts, they all stemmed from a version of this type of grant. Apologies, I need a minute to let my family members in. Okay, let's hope this works. Okay, so as I was saying that we've been funding fellowships since 2021. We've put in a whole bunch of money towards different types of fellowship opportunities, and most of these researchers have been working on therapeutic screening and development, translational research, as well as assay development for biomarkers and the like. And you can see the levels of funding that we actually provide. And this has been instrumental in getting us to the position that we are. And I'm hopeful that, in the years forward as well, we will be able to support a strong cohort of fellowships going forward. All of these researchers that you see here were supported in 2024 through our funds. They work on a mix of nanoparticles, gene therapies, early basic organoid work, which will inform future gene therapies and the like. And so I'll end on really, plug to you, the community over here, that it would be great if you can support our efforts by participating strongly in our Holiday Heroes fundraisers as you have done in the past. And I'll stop there and maybe take some questions.